So, hello students, welcome back to CBSE Vedantu Solutions, where we do NCERT line by line of all the chapters. And today's chapter is very unique and very basic. So, when I say basic, I mean is that this chapter right here is the foundation chapter for all the other chapters in your class 11th and 12th. So, if you ask me which is the first chapter which you need to master before moving on to the other chapters, I would say cell the unit of life. Because whatever you study in this chapter will be the foundation in principles of inheritance, molecular basis, cell cycle and cell division, and even the reproduction chapters. So let's master this class, and hopefully we understand every single line of NCERT. And after this class, you will not even have a single doubt regarding the cell, the unit of life. Okay, so let's start right now, students. The first NCERT line by line will go. Okay, we'll go NCERT line by line, and hopefully there is no doubt. Anytime you have a doubt, let me know in the comment section and I will reply to it. Not a big deal, right? So let's start reading the NCERT line by line. Cell the unit of life. Cell the unit of life. See, when you look around, you see both living and non-living things, obviously. So if you look around, so right now I do not have any very massive non-living things, but I'm alone sitting here, but I do have certain non-living things. Non-living book, non-living. I have certain, like a remote, non-living. But there are certain microbes around me, right? There are certain microbes around me that are living. In the corner, I can see a spider that is living, right? That is going to be living in nature. So that is what means the first line. You can see living and non-living things around you. Now you must have wondered and ask yourself, what is that makes an organism living? And what is it that inanimate things does not have, which a living thing has? Obviously, the main difference between living things and non-living things is what? The main difference between living things and non-living things is that in the case of living things, it is made up of cells. It is made up of cells which gives us the life. Right? Now, the answer to this is the presence of basic unit of life, that is the cell in all living organisms. So if you want me to tell if something is living, it should be made up of cell. A single cell or a multi-cell, that can vary, right? All organisms are composed of cells. Some are composed of a single cell, and we call them unicellular in nature. Yes, we call them unicellular organisms if they're made up of a single cell. Now, if they're made up of multiple cells, students, if they're made up of multiple cells or many cells, we call them multicellular organisms. Multicellular organisms. Multicellular organisms. Any doubt in the first paragraph? Quickly ask me, students. Any doubt? There should be no doubt. It's fairly simple. The first paragraph, right? Very simple paragraph. Now let's go to the next one. Next paragraph. Now, next thing you need to understand, what is cell? All of you will tell me, sir, cell is the basic unit of life. Basic unit of life. Or you will also say me, sir, cell is a fundamental unit of life. Both are correct. But let's read what the NCERT says. First of all, NCERT is talking about unicellular organisms. Organisms with a single cell, right? Unicellular organisms are capable of independent existence and perform essential functions of life. That is, unicellular organisms are able to self-sustain. They're able to take care of their needs. Independent existence, they do not have to depend on other organisms. And perform all the essential functions of life. That is, deriving the nutrition, digesting the nutrition, and releasing the waste material. Essential functions of life. And also reproduction. Right? Anything less than a complete structure of a cell does not ensure independent living. That is, basically, if it is not complete, it is not independent. Now, hence, a cell is fundamental structural and functional unit of all living organisms. That is, every single organism is made up of, structurally, the organism is made up of multiple cells or a single cell. Now you need to understand a few things here, right? Something called as, there are a few scientists, we'll encounter a few scientists right now. One of the scientists called as Anton von Leeuwenhoek. Anton von Leeuwenhoek. Okay. Now before we go to Anton von Leeuwenhoek, can anyone tell me in the chat who was the scientist who observed the first cell? Tell me, who was a scientist who observed the first ever cell? Like he was a scientist who was like, he looked under the microscope or a simple microscope here, or he looked into something and he was able to tell, yes, this right here is a cell. Who was a scientist? Can anyone tell me the name of a scientist who gave the first cell? That is nothing but your Robert Hooke. It's going to be Robert Hooke. Robert Hooke was the first scientist. Robert Hooke was the first scientist to describe the cell. Robert Hooke. It's Robert Hooke. But tell me, when Robert Hooke discovered this cell, right? Robert Hooke discovered a cell. Was it living cell or dead cell? Take your time and tell me. Was it a living cell or was it a dead cell? So you obviously tell me, sir, we have learnt in previous classes, Robert Hooke discovered a cell, but it was a cork cell, right? And the cork cells are dead cells. Cork cells are dead cells. So you will tell me, sir, Robert Hooke discovered a cell, but it was a dead cell. It was a dead cell. Then tell me, who was the scientist who first observed the living cell? The first living cell was observed by Anton von Leeuwenhoek. That is a living cell. Please know the difference between the two discoveries. One discovered the living cell, one discovered a dead cell. Okay, next, one more scientist is called as Robert Brown. We have Robert Brown. Okay, Robert Brown discovered the nucleus. He discovered the nucleus. So three different scientists: Anton von Leeuwenhoek, Robert Hooke, Robert Brown. Three different scientists names you need to know. Okay, thank you so much. Thank you. Happy this day. Yes, thank you so much. Three different scientists you need to know. Three different scientists. Now, next we'll go. Robert Brown later discovered the nucleus. The invention of microscope and its improvement led to the electron microscope, revealing all the structural details of the cell. So once electron microscope was discovered, all the internal organelles became clear. Internal organelles of the cell was clearly visible now onwards. So after the discovery of electron microscope, obviously if you have electron microscope, you can look inside the cell and tell me, sir, this is Golgi body, we have ribosomes, we have the endoplasmic reticulum. All that can be visible only under the electron microscope. Only visible under the electron microscope. Okay. Now students, next concept is called as cell theory. Any doubt till now? Quickly ask me students in the chat. I will look at the chat now. Any doubt till now? Any doubt till now? Thank you for happy this day. Yes. Any doubt till now? No doubt. Beautiful. Okay. You're giving me celery. Okay, celery my gift for you, special this day. Okay. Thank you for the celery. Okay. So next we'll learn something called cell theory. So students, you need to understand, in cell theory there are three main scientists. There are three main scientists in the cell theory. Okay. Now first, let me write it for you, then we will read what is given in NCERT. The first scientist is called as Matthias Scheiden. Matthias Scheiden was a German botanist. He was a German botanist. Okay. He was a German botanist. First scientist. Then we have second scientist. That is Theodor Swan. Theodor Swan was a British zoologist. Okay, British zoologist. Now, third one I'll tell later on, but understand the first two scientists, what do they do? First, this one, the German botanist students, the German botanist or botanist right here. His main work was on plants. Right? His main work was on plants. He studied different kinds of cells. In the case of plants, and he also told us these different kinds of cells will make up. Different kinds of tissues. Cells will make up the different types of tissues. That was by Matthias Scheiden. Matthias Scheiden was a German botanist. He studied plants and he told us that different plants make up the different type of cells and different type of cells make up the different type of tissues. That's all. That's all. Now, what does Theodore Swan told us? Theodore Swan did three main things. The first thing he discovered was the plasma membrane. Plasma membrane. See, back then he did not know it was plasma membrane. He told us that beneath the cell wall there is a separate layer called as plasma membrane. Okay. Today we know it as plasma membrane. Back then he did not know. Okay. The second thing he noticed that was cell wall. Cell wall is only found. It is a unique character. Cell wall is a unique character. Unique character to the plants. It is a unique character to the plants. Animals do not have the cell wall. Animals do not have the cell wall. Cell wall can be only be found in the case of plants. Okay. The third thing he told was, along with the Matheus-Seidel, he told us that animals and plants composed of cells and product of cells. So all the plants in the world, all the animals in the world are made up of cells and a product of cells. This was given by Theodore Swan. So together they gave the cell theory. But in this entire cell theory, students, they could not explain how the cells came into existence. They could not tell how the cells came into existence. So for that, there was one more scientist. There was one more scientist called as Rudolf Virchow. He came into existence and he told us that cells come into picture, cells arise from a pre-existing cells. That is Rudolf Virchow, the third scientist. The third scientist is Rudolf Virchow. Rudolf Virchow in the year 1855. In the year 1855, he first explained. One second, let me use the color. First time explained. What did he explain? He explained us that cells divide and new cells are formed from pre-existing cells. So all the cells which are present in the world are from the pre-existing cells. Okay. Like mitosis will happen only when there is one cell. Like one cell will divide into two cells. That is mitosis. So one cell is dividing into two cells. That phenomenon was told by Rudolf Virchow. Together, the Matheus-Seidel, Theodore Swan and Rudolf Virchow, all three scientists together gave the cell theory. And cell theory says that there are two main points of cell theory. Okay, let's read what is given here, then I'll tell you the two main points of cell theory. See, it says that Matheus-Seidel, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells. I told you, different kinds of cells, which form tissue of the plant. At the same time, Theodore Swan, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer, which is today known as plasma membrane. See, first thing was what? Plasma membrane. Thin outer layer. There's a thin outer layer called as plasma membrane. Okay. He also concluded, based on his studies on plants. See, he also worked on plants also. On plants, tissues, that the presence of cell wall is a unique character to the plant cell. See, unique character to the plant's cell wall. Next point. On the basis of this, Swan proposed hypothesis that bodies of animals and plants are composed of cells and a product of cells. Animals and plants composed of cells and product of cells. Three main points by Theodore Swan. Now, Seidel and Swan together formulated a cell theory. Now, this theory did not explain. What did it explain? It did not explain how new cells were formed. Like how the new cells are coming into existence at all. It could never explain all that. So to explain that, we got a new scientist called as Rudolf Virchow, right? Rudolf Virchow. He came to existence and he told us that he first explained that cells divided. Cells are dividing. Cells divided and new cells are formed from the pre-existing cells. See here. He first explained cells divide and new cells are formed from the pre-existing cells. And he called this like a magic spell, one nice spell he told. He told us that omnis cellula e cellula omnis cellula e cellula, that is cells come into existence from the pre-existing cells. Okay, pre-existing cells. Now what is the final cell theory, students? Shannon's one cell theory was what? All organisms are composed of cells and product of the cells. First point. All cells arise from the pre-existing cells. Second point. The two main points about the cell theory. So with that, cell theory is over. Now you need to understand there are few organisms which do not follow cell theory. For example, a virus, a heart cell, a neuron. All of this do not follow the cell theory because they do not divide, right? They do not divide at all. So with that, the first part is over. Very easy, right? Every line of NCERT we are covering. Every line of NCERT we are covering and going. Now we'll go to the overview. Ready? Ready, students? Overview of cell theory. Friends, take this opportunity, right? This channel is dedicatedly created for all the students who want to not only crack NEET but also want to score really well in the CBSE board exams. And the only way you can score really well in the board exams is when you are very thorough with your CBSE or NCERT textbook. Then only you get really good marks. And same goes for NEET students, right? Unless they master the NCERT, read the NCERT 3-4 times, right? You'll never get that confidence. So take this opportunity to learn more. What is harm in learning more, right? And it is free of cost, right? Now let's read the overview of cell theory. Okay, overview of cell theory. So before we go to the overview of cell theory, students, tell me, do plant cell have cell wall? Do plant cell have cell wall? Yes. Plant cell have cell wall. They will have cell wall. Will they have cell membrane? Yes, they will have cell membrane. They will have cell membrane present. Cell membrane also is present. Okay. Now, where do you think nucleus is, students? Nucleus is obviously in the nucleus. What do we get in the nucleus? Within the nucleus, we have the chromosomes. Within the nucleus, we have the chromosomes. And within the chromosomes, do we have DNA? Yes, we have the DNA. Yes, we have the DNA within the chromosomes. Okay. And nucleus is present inside the cell, like a brain of the cell. Brain of the cell. Okay. Now, one more thing I'll tell you here is a particular organism, right, which does not have cell wall. This has been asked many times a question, and you will learn it in biology classification also. There's an organism called as mycoplasma. Mycoplasma, which lacks cell wall. It lacks the cell wall. Lacks cell wall. Remember this. Okay, it might be asked somewhere. It'll be asked. Let's read overview. You have earlier observed the cells in an onion peel or human cheek. Class 6, 7th, 8th you studied. Okay. The onion cell structure, which is typical plant cell, has a distinct cell wall. Yes, plant cell has a cell wall in its outer boundary, and just within it is the cell membrane. So there is also presence of cell membrane. Okay. The cells of the human cheek have an outer membrane and as a delimiting structure of the cell. Tell me, students, in the case of animal cell, which is the outermost layer? Outermost layer in the case of animal cell is nothing but the plasma membrane. But in the case of your plant cell, it is the cell wall. Cell wall is the outermost. Cell wall here is the outermost layer. Okay. Now, inside each cell is a dense membrane-bound structure called as nucleus. Inside you have a nucleus. Now this nucleus contains chromosomes, I told you, contains chromosome, which in turn contains genetic material, that is the DNA. DNA is present. Cells that have membrane bound nuclei are called as eukaryotic cell. And very important point coming up now. Okay, let's take one separate slide to answer this, and then we'll come back here. So entire cell, students, entire cells can be divided into three categories. Entire cells can be divided into two categories. Okay, the first one is called as prokaryotic cell. Prokaryotic cell. The second one is called as eukaryotic cell. Eukaryotic cell. Prokaryotic cell will have what? They will have nothing actually. They'll not have no membrane bound organelles. No membrane bound organelles. There is no membrane bound organelles in the case of prokaryotes. Now what about the case of eukaryotes? Eukaryotes have membrane bound organelles. They have membrane bound organelles. Now what is the meaning of this membrane bound organelle, students? The meaning is very simple. It could be endoplasmic reticulum, nucleus, Golgi body, lysosomes. All of these structures have a separate membrane around it. They have membrane around it. In the case of prokaryotes, they do not have any membrane. There is no membrane at all. Okay, vacuoles, microbodies, everything. But students, tell me, under eukaryotes, do we also have something called as non-membrane bound organelles? Just like prokaryotes, under eukaryotes, do we have something called as non-membrane bound organelles? Do we have? Yes, we have. There are two organelles as such. Even though they are found in the eukaryotic cell, they do not have any membrane. The first one is called as your ribosomes. And the same ribosome is also found in the case of prokaryotes, remember, ribosomes. And the second one is called as centrosome. Ribosome is found in both plants and animals. But centrosome is only found in the case of animals. Centrosome. Okay. Non-membrane bound organelles in the case of eukaryotic cell. Okay. Now let's read what is given here, students. Whereas cells that lack a membrane bound organelle nucleus are called as prokaryotes. See, both prokaryotes and eukaryotes, a semi-fluid called as cytoplasm occupies the volume of the cell. Okay. So what is the common thing between them? Let's not common between them. Common things between prokaryotes and eukaryotes. Tell me. Both prokaryotes and eukaryotes have a cell membrane. They have a cell membrane on top which covers it. The cell membrane. They have cytoplasm. Semi-fluid thing. Cytoplasm is there. Yes. What about the ribosomes? Ribosomes are also common. Yes. Ribosomes are also common. Both will have DNA. They will have DNA, but the DNA structure will be different. But DNA is present. Okay. Now let's read what is given in NCERT. The cytoplasm is the main area where the cellular activities in both plants and animal cells. Okay. Various chemical reactions occur to keep the cell in a living state. So chemical reactions are taking place in the cytoplasm. Glycolysis example. Beside nucleus, the eukaryotic cell have other membrane bound organelles like your endoplasmic reticulum, Golgi complex, lysosomes, mitochondria, microbodies, vacuoles. All of them, students, will have membrane. Why? Because they're eukaryotes. Right. The prokaryote cells lack such membrane bound organelles. See, I told you, no membrane bound organelles in the case of prokaryotes. Ribosomes are non-membrane bound organelles. See, ribosomes are what? Under non-membrane bound. They're under Non-membrane bond. Okay. Ribosomes are non-membrane bond organelles found in all cells, both eukaryotes as well as prokaryotes. See, I told you, they're present in eukaryotes, also present in prokaryotes. Here, right here. Okay. Prokaryotes. Within the cell. Ribosomes are not found only in the cytoplasm, but also are found in two organelles. Where do you find ribosomes, students? Ribosomes are also found in where? Where do you find ribosomes? Ribosomes can be found in the cytoplasm. It can be found in the case of your chloroplast in the case of plants, mitochondria, and also rough endoplasmic reticulum. All these four places: cytoplasm, chloroplast, mitochondria, and rough ER. In all these four locations, we can find ribosomes. Okay. Now, animal cells contain another non-membrane bond organelle called as centrosome, which help in cell division. See, centrosome is here. The main function is cell division. Main function is cell division. Beautiful. Now, next thing is the cell size. Okay. Students, remember, the cell size can always vary. Students, do you want this PPT? Tell me. If you want this present, do you want this notes for this one also? Do you want notes? Notes I'll be giving it on my Telegram channel. Whatever I'm writing, if you want my reference, of it, I'll give it in Telegram channel. All this. I think I'm writing a lot, right? I am writing a lot. That we are making some side by side. We are making some notes, right? Side by side, we are making notes as we write also. I think as we learn. So if you want, I will give you these notes also. You can download these notes also. Simple notes it are. They are simple notes. Now, cell size. So cell size can vary in the case of plants. In the case of organisms, cell size can vary completely. For example, tell me the smallest cell. Smallest size. Yesterday I told take notes because yesterday we wrote less. Today we are writing a lot. But take notes, students. What is wrong in taking notes, right? What is wrong in taking notes? You can take the live notes. And you can also write with me, right? So if I'm writing something, I want you to write it in a small book or the sticky notes, right? Take a small sticky note. Next to your book only you can keep it. Or you can stick it on the NCERT only. Okay. Now, tell me the smallest size. That is going to be the mycoplasma. That is roughly around 0.3 micrometer. Now tell me the largest now. Largest cell. Largest cell is going to be ostrich egg. Largest cell is going to be the ostrich egg. Okay. Now tell me, what about in the case of a simple bacteria? What is the size of a bacteria? Size of bacteria is between 3 to 5 micrometer. Bacteria. Now what about human RBC? Human red blood cell. Human red blood cell is around 7 micrometer. A little bit bigger than bacteria. That's why bacteria can enter into the cell. Right, human cell, human RBC. Last one is longest cell. See, not largest, longest cell. Longest cell is basically the nerve cell. So all of these points you need to remember. They can ask you question based on arrangement. They can give you all the cells. They'll ask you to arrange them here and there. Okay, arrange them. 7 micrometer. Okay, 7 micrometer. Now let's read what is given in NCERT, all of you. Let's read what is given in NCERT now. The cells differ greatly in size, shape and activity. For example, mycoplasma is the smallest cell, 0.3, wrote here. length, while bacteria is around 3 to 5, wrote here, right? Largest is ostrich egg, done. Where is ostrich egg? Here. Red blood cell is around 7 micrometer, done. Nerve cell is the longest cell, done. See, next time when you read this part, right, if you just read this one paragraph, you're good to go. You don't have to read the entire thing here. Cells also vary greatly in the shape. For example, look at the shapes. We have biconcave shape, biconcave, amoeboid shape, like diapedices movement they show, columnar, like be around columnar epithelium cells, branched nerve cell, tracheids, elongated cells, the mesophyll cells, green color with chloroplast, green color cells with chloroplast, oval shape, right? Now, if you look at the trachea, students, look at the end of tracheids now. Ending of tracheids is always tapering end. See, you see, tapering end, tracheids. But vessels do not have tapering ends. Vessels will never have tapering ends. So that is one of the major difference between tracheids and vessels. Tracheids tapering end, vessels do not have tapering ends. Okay. Next one. Now we will start with prokaryotic cell, now, students. Now we will start with prokaryotic cell. Any doubt till here? Tell me. Any doubt till here? Quickly ask me. If there is no doubt, we'll start with prokaryotic cell now. Can we start? Prokaryotic cell. Vessels will never have tapering ends. In fact, vessels will be made up of vessel elements. They have perforated end plates. Okay, remember that. Tapering ends only in the case of tracheids. So in examination, if they give you a diagram, you should be able to tell what is tracheids. Okay, how are tracheids? The beast, what should I repeat, sir? Beast, sir, tell me. Tracheids, I told you, tracheids will have tapering end. This is called as tapering end, pointed ends. You see, pointed ends. Okay, very easy. Now prokaryotic cell. First of all, where do you find prokaryotic cell? Simple question. In which organisms do you find prokaryotic cell? Where do you find prokaryotic cell? Tell me. Where do you find prokaryotic cells? Prokaryotic cell, obviously, when you think of prokaryotic cell, the kingdom which should come to your mind, tell me. Which kingdom should come to your mind? From the 5 kingdom classification, which kingdom should come to your mind? The first kingdom called as kingdom Monera. The kingdom Monera should come to your mind. Kingdom Monera. Protista are eukaryotes, unicellular eukaryotes. But kingdom Monera is prokaryotic in nature. Now, under kingdom Monera, you should always think of first is bacteria, the famous ancestor that is bacteria. Stupid bacteria should come to your mind first of all. Apart from bacteria, you should also understand or remember blue-green algae. Blue-green algae. You should always remember mycoplasma. Mycoplasma. And also PPLO. Pluro, pneumonia like organisms. This is all this should come to your mind when think of are simple prokaryotic organisms. Okay, prokaryotic organisms. All of these organisms should come to your mind. Okay. Now let's read what is given in NCERT now. Okay. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO. Okay. They are generally smaller and multiply more rapidly. For example, see, let me write it for you here so you will understand the properties. Properties here. They have smaller size. And they multiply rapidly. Quickly, rapidly. 20 minutes, one new cell is there. Every 20 minutes, new cell is there. Bacteria. Every 20 minutes, E. coli. Now, what could be the basic shape? Basic shape is very important to understand. What are the basic shape of these cells? Basic shapes can vary, okay? For example, it can be bacillus. It can be bacillus, which means rod-like. Then we have something called as Vibrio. Vibrio is comma shape. Then we have something called as coccus. Coccus is nothing but spherical. Just spherical. Then students, we also have spirulum. I have spirulum, which is basically spiral shape. Spiral shape. See this. Can you see? This is shapes. It's not given now. It's fine. Okay. So let's read what is given here now. The four basic shapes of bacteria is bacillus, rod-like, coccus, spherical, round, vibrio is comma-like, yes, and spirulum is spiral shape, like spiral shape. Organisms of prokaryotic cells are fundamentally similar, even though prokaryotic cells exhibit wide range of shape and function. They are similar because they are prokaryotic. How much they can even vary, right? How much they can even vary. Now, students, tell me first of all, do plants have cell wall? Yes. Plants have cell wall. They have cell wall. What about cell wall in the case of bacteria? Do you think bacteria has cell wall? Yes, they have cell wall. In the case of plants, the cell wall is made up of cellulose. In the case of bacteria, the cell wall is made up of peptidoglycan. Peptidoglycan. Okay, peptidoglycan. It is made up of peptidoglycan in the case of bacteria. Peptidoglycan. Okay, except mycoplasma. Mycoplasma do not have any peptidoglycan. Always remember that. Okay. Now, do they have cytoplasm? Yes, they have cytoplasm. Do they have nucleus? Well-defined nucleus? No. No well-defined nucleus. No well-defined nucleus. Not there at all. Not there at all. Okay. Do they have genetic material? Genetic material? Prokaryotic cell? Yes, they have genetic material. Yes, they have genetic material. They have something called as, they have single chromosome that is mostly circular in nature. Circular DNA. This circular DNA is called as plasmid. Extra chromosomal DNA, they have something called as plasmid. Plasmid they have. They have the plasmid. Nuclear envelope is absent. Any membrane bound organelle, everything is absent. Okay. Do they have ribosomes? Yes, they have ribosomes obviously. 70 is ribosomes. 70 is ribosomes. They have ribosomes which are 70 is ribosomes. Okay, 70 is ribosomes. Prokaryotic cell have cell wall surrounding the cell membrane except in mycoplasma. PYQ. The fluid matrix is filled in the cell cytoplasm. So there is cell wall is present, cytoplasm is present. There is no well-defined nucleus. No well-defined nucleus. Genetic material is basically naked. Genetic material is naked. Okay. Not enveloped by any membrane. No membrane at all. In addition, genomic DNA, the single chromosome or circular DNA, many bacteria have small circular DNA outside the genetic material, that is outside the genomic DNA. These small DNA are called as plasmid. So they also have, apart from the main DNA, they also have the plasmid. So they have main single chromosome or circular DNA. They also have a small DNA, that is plasmid. A plasmid DNA confers certain unique properties, phenomena to bacteria. For example, you'll study more about your plasmid, exploitation of plasmid. You cut the plasmid, you add something to the plasmid, you modify the plasmid. All that you'll study in the chapter biotech. Okay, biotech. Yes, biotech. Now, one such character is to resistance to antibiotics. In higher class, you will learn. Okay. Nuclear membrane is found in eukaryotes. No organelle like the one in the eukaryote are found in prokaryote except the ribosomes. Ribosomes are same. Eukaryote, prokaryote, ribosomes are same, no membrane bound. But in eukaryotic it is 80s, prokaryotic it is 70 years. Pro 70 years. Eukaryotic 80 years. Simple difference. Prokaryotic have a something unique in the form of inclusions. There is something called as inclusion bodies. A specialized unique in form of inclusions, a specialized differentiated form of cell membrane called as mesosome is present here. For example, students, I'll teach you something very special now. Just draw with me. All of you draw. Time to draw. Time to draw now. One second, students. Let me go to more. So a particular bacteria has always 3 different layers, students. A bacteria always has 3 different layers. Full protection it has. A bacteria has 3 different layers always. The first one is the outermost layer. The outermost layer is called as glycocalyx. This outer layer glycocalyx can be present in the form of loose sheet, or it can be present in the form of thick and tough layer. When it is present thick and tough layer, it is called as capsule. Loose sheet, it is called as slime layer. Okay. Now second layer, friends. Second layer is basically the cell wall. What is cell wall made up of, friends? I told you already. Cell wall is made up of peptidoglycan. That is the cell wall made up of peptidoglycan. All of you know that. Okay. Now, what is the innermost layer? Which is the one second. Second layer. Now I have the innermost layer. Innermost layer is the plasma membrane. Plasma membrane is the innermost layer. This plasma membrane is selectively permeable. It is going to be selectively permeable. It doesn't allow everyone inside. It doesn't allow anyone or everyone on the inside. Okay. Now, 3 layers done. Now we learned just now in the foldings of the plasma membrane, there is some extensions or foldings. There are some extensions or foldings in the plasma membrane. That extensions or foldings in the plasma membrane is called as mesosomes. It is called as your mesosomes. Okay, mesosomes. Then students, then there can be certain types of mesosomes which can hold some pigment or color, like chlorophyll or something. A pigment, it can hold a pigment. Such type of structures which can hold pigment is called as chromatophore. This chromatophore is seen in the case of blue-green algae. In the case of your blue-green algae, chromatophore. Okay. Do they have ribosomes? Yes, they have ribosomes. They have the ribosomes. Do they have vacuoles? Some amount of vacuoles? Yes, they have vacuoles. Now, apart from that, from the plasma membrane here, there is some form of structure arising. Can you see? Plasma membrane, some structure is arising outside. Like a tail-like structure, like a whip. The structure is arising right here. This structure is called as the flagella. Flagella helps in motility. It helps in the movement of the bacteria. It helps in movement of the bacteria. That is flagella. Now, apart from flagella, there are two other appendages. There are two other appendages coming out of this. There are two other appendages from the body of the cell. These two appendages are called as your, it can be pilae or fimbriae. Pilae or fimbriae. Now, do pilae and fimbriae also help in movement? No. Pilae and fimbriae do not help in motility. In fact, pilae and fimbriae help in attachment. They help in attachment. So this is the entire structure of your bacteria. Apart from this, you will have inclusion bodies and everything is there, but now they are not necessary here right now. Okay, this is the entire structure of bacteria. Now, what did we learn till now? Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. Cell envelope consists of tightly bound three layers. Told you. The outermost is called as glycocalyx, which can be in the form of two things. Glycocalyx can be in the form of tough and thick, called as capsule, or loose sheet in the case of your slime layer, right? Followed by cell wall, and plasma membrane is the innermost layer. Although each layer has envelope, each of the layer of the envelope performs distinct function, they can act together as a single productive unit. They act and perform. Bacteria can be classified into two groups based on the difference in the cell wall, envelope, and manner in which they respond to the staining procedure. So basically, if they have high peptidoglycan or low peptidoglycan, based on the envelope, bacteria can be divided into two things. If it has thick peptidoglycan, it is called as gram positive, which can take up the stain. Thin peptidoglycan cannot take the stain, that is called as gram negative. Thin peptidoglycan layer cannot take the stain, that is called as gram negative bacteria. This is called as the foundation of differences, foundation of making differences, classification. Okay. Sure. Slime layer, capsule. The cell wall determines the shape of the cell and provides strong structural support to prevent bacteria from bursting into collapsing. Plasma membrane is selectively permeable in nature and interacts with outside world. This membrane is similar to structure of eukaryotes. That is the plasma membrane. Okay. A special membrane structure is called as mesosome, which is formed by the extension of plasma membrane into the cell. Can you see here? Mesosomes. Extension of the plasma membrane. Extension of plasma membrane. This extension can be in the form of vesicles, tubules, lamella. Now, functions are very simple. This is very important now. What is the function of the mesosomes? Their first function, can you see? First one is replication of DNA. Second one is distribution of daughter cells. Third one is respiration. Fourth one is secretion. Fifth one is increasing surface area. And sixth one is the enzymatic activity. 6 different properties of mesosomes. Anyone can be asked a question. Let me tell you only. 6 different properties. Okay. In some prokaryotes like cyanobacteria, there are other membranous structures into the cytoplasm called as chromatophores, which contains pigment. See, cyanobacteria, chromatophores. Cyanobacteria, blue-green algae, chromatophore. Now, bacterial cells may be motile or non-motile. If motile, they will have the flagella, thin flagella. Bacteria show a range of numbers and arrangement of flagella. The bacterial flagella is composed of 3 parts, that is basal body, hook, and filament. Basal body, hook, and filament. 3 different parts of a particular structure of the flagella. The filament is the longest portion and extent in the cell surface with the outside. Besides, flagella, pili, and fimbriae are also structural structures of the surface structures of the bacteria, but do not play in the role of motility. The pili are elongated tubular structures made up of special protein. The fimbriae are small bristle-like. Bristle means like brush. Bristle-like fibers sprouting out of the cells. In some cells, they are not only to help the attached bacteria to the rock, they are also in the host tissue. Okay, attached to the host tissue. So, with the help of pili and fimbriae, a bacteria can attach to the host. Attach to the host. Ribosome inclusion bodies. In prokaryotes, ribosomes are associated with plasma membrane, all of you know. So we have 70 S ribosome in the case of prokaryotes, which is divided into 50 and 30 S. You will tell me, sir, 50 plus 30 is 80, but why giving 70? Because this is based on biology, that is sedimentation coefficient, not an addition. Okay. Ribosomes are a site for protein synthesis. Several ribosomes may attach to a single mRNA. See, this is a single mRNA. Many ribosomes can be attached to this. Many ribosomes can be attached to this. Okay. Such mRNA, which has many ribosomes attached to it, is called as polysome or polyribosome. Poly means many, ribosomes. Okay. Ribosomes form a polymer. Polysome translate to the RNA into protein. Last concept for prokaryotic is inclusion bodies. Reserved material in prokaryotic cell are stored in the cytoplasm in the form of inclusion bodies. So inclusion body is a storage unit. Since vacuoles are not very developed, they have their own fridge. These are not bound by any membrane bound or system and live in the cytoplasm. Example: phosphate granules, cyanophesian granules, glycogen granules. All these granules are basically examples of inclusion bodies. Inclusion bodies are what? They are fridge. All the nutrients, everything is stored inside the fridge. Gas vacuoles are found in the blue, green, and purple photosynthetic bacteria. Gas vacuoles are also present in certain bacteria. Okay. Now, students. Now we will take one 10 minutes break and we'll come back and we'll do eukaryotic. We'll do 15 minutes break. I'll go drink some water and we'll take a 10 minutes break. It's fine. Now break till 6:13. Break till 6:13. Then we'll do eukaryotic cell. Then we'll do eukaryotic. We'll cover today. We'll cover till I can fill the chapter also if you want. I can sit here aramse for 3 more hours and teach. If you're ready to listen, that is. Okay. I don't mind teaching the entire chapter, but I think we will stop at endoplasmic reticulum. We'll finish endoplasmic reticulum or we'll finish Golgi complex, I guess. No. Let's finish at ER. Let's finish at ER today. Okay, let's finish at ER. Let's make it 15. 6:15 only will come off. It sounds like a proper number. 6:15. Okay. A little tired today. That's all. A little tired. Fatigue. So students, welcome back. Welcome back to the session of the cell by cell, line by line, cell the unit of life. That is cell the unit of life. Now we'll be starting off with the eukaryotic cell. Quickly let me know in the chat if you're ready for the eukaryotic cell right now. Let me know in the chat quickly, all of you. Yes, eukaryotic cell. Can we start? Can we start, students? Eukaryotic cell. Yes. Yes. Let me know if we can start. Can we start? Eukaryotic cell. Now, first of all, tell me, students. First of all, tell me, eukaryotic cell. In which organism do you find eukaryotic cell? Quickly tell me. In humans, yes. In humans, we find eukaryotic cell. And humans belong to kingdom Animalia. So, can I write animals? Can I write animals now, students? Tell me. Animals. Yes. In animals, we find eukaryotic cell. Okay. Now, what about protist? Protist. Yes. In protist also we find eukaryotic cell. What about in the case of plants? Yes, we find eukaryotic cell. And also in the case of fungi, also we find eukaryotic cell. So, except Monera, which is the kingdom where we find only unicellular or prokaryotic cell, except Monera, all the other kingdoms are eukaryotic in nature. Is that point clear to all of you? Tell me. Is that point clear to all of you? Yes. Now, tell me some characters. Tell me some characters of eukaryotic cell. Obviously, the first character is going to be all the eukaryotic cell are membrane bound. Yes, they are going to be membrane bound. They are going to be membrane bound organelles. Membrane bound organelles. All the organelles will have separate membrane around them. Membrane bound organelles. The second thing is called as the nuclear envelope. It's very special because the nucleus is well defined. The nucleus is well defined and there is nuclear envelope here. Nuclear envelope. Apart from that, a eukaryotic cell will have a separate framework. The framework or the backbone of a cell is called as cytoskeleton. It's called as what? Cytoskeleton, and the cytoskeleton will be present now. That is called as complex locomotory and cytoskeletal structures. Cytoskeletal structures. Complex locomotory. For example, there could have cilia. Cilia is there. Very complicated cilia. We have contractile vacuoles are there. So you learn all that in the under eukaryotics now. Under eukaryotics. Right. Now, let's see what is given here. Let's read what is given here. Obviously, all of you know, in the case of plant cell and animal cell, both are different. We'll get to that later on. We'll tell you here only. Wait. Tell me, in the case of plant cell and animal cell, simple difference. Simple difference. Here, in the case of plant cell, it will have cell wall. Yes. The cell wall will be absent in the case of animals. The plant cell will have something called as plasters. It will be absent here again. Plant cell will have large central vacuole. Large central vacuole. Here, small vacuole. There is going to be small vacuole. Small vacuole. Okay. There will be small vacuole here. There will be small vacuole here. Okay. Small vacuole. Now, in the case of animals, they have separate structure called a centriole also. Or centrosome. Centrosomes are present in the case of your animal cell. Centrosomes. Okay, also present in the case of animal cell. Okay, let's read what is given here now, students. Eukaryotic cell includes all protist animals, plants, animals, and fungi. Yes, included. In eukaryotic cell, there is an extensive compartmentalization of cytoplasm through presence of membrane-bound organelles. What is compartmentalization? Is that within one cell, every single part is divided into separate compartments. Like how in a train we have a compartments, right? Similarly, in a cell, there are multiple different compartments. Eukaryotic cell possess an organized nucleus. See, well-defined nucleus, right? Well-defined nucleus with a nuclear envelope. Done. In addition, eukaryotic cell have variety of complex locomotory and cytoskeletal structures. Okay. Genetic material. Tell me, what is genetic material here? How is the genetic material? Tell me, genetic material. In the case of eukaryotes, genetic material, students, genetic material is properly well defined. It is present in the form of DNA, right? It is present in the form of DNA, genetic material, right? Genetic material, where is it? Organized into chromosomes. Organized into chromosome. Proper chromosomes can be seen here. Okay. All eukaryotic cells are not identical. Plant and animal cells are different. As the former possess cell wall in the case of plant cell, plastids in the case of cell, and large central vacuole in the case of plant cell. Okay. Which is absent in the case of animals. On the other hand, animal cells have centrioles. See, centrosome is made up of 2 centrioles, right? Centrioles, which are absent in the case of plants. So, centriole is absent in the case of plants. Basic difference between plant cell and animal cell. See, if you look at the diagram also, you can tell here. See, this right here on the left side, large central vacuole. So, plant cell, right? Plant cell. Can you see cell wall? Plant cell. Then we also have the where is it? Chloroplast. In the case of your plant cell. In the case of plant cell. Now, what about in the case of animal cell? In animal cell, you will have the distinguishing feature is going to be the centrioles. Here, centrioles is going to be the distinguishing feature in the case of animal cell. So, you should know the proper difference between a plant cell and an animal cell. Okay. Yes, Natasha, bacteria also has cell wall. Can anyone tell Natasha that, but what is the difference? What is the difference between a plant cell wall and a bacterial cell wall? Can anyone tell Natasha? What is the difference between a plant cell wall and a bacterial cell wall? Tell me quickly. What is the difference? Now we'll go to the cell membrane. Now we'll go to the complete cell membrane. Quickly, all of you. What is the cell membrane made up of? Okay. Friends, remember, the other name of cell membrane is also called as plasma membrane. It is also called as plasma membrane. It is also called as plasma membrane. Okay, plasma membrane, all of you remember that. The detailed structure of membrane was studied only after the evident electron microscope in the 1950s. So before 1950, no one knew about plasma membrane. Plasma membrane can only be studied very well when the electron microscope was discovered. Meanwhile, chemical studies on the cell membrane, especially in the RBC, enabled the scientists to deduce the possible structure of plasma membrane. So due to some chemical reactions, they were able to tell, yes, this is how the plasma membrane looks like. Okay. But they did not exactly know how the plasma membrane is, what exactly is happening, the structure of plasma membrane. But they did not know that till then. Okay. These studies showed that the cell membrane is mainly composed of lipids and proteins. Students, tell me. Shobharatani. Okay. I'm very bad at names. All of you know that, right? So tell me, what is cell wall made up of? Cell wall is made up of your lipids. It is made up of proteins. Lipids is generally around 20 to 70%. Yes. It can vary between that. Protein also can vary between 20 to 70%. With varying degree can be there. Apart from that, we also have carbohydrates. We have the carbohydrates. Carbohydrates are just 1 to 5%. That's all. 1 to 5 carbon. Now, students, anything is missing. Lipid is there, protein is there, carbohydrate is there. Is anything missing? Now tell me. Anything is missing? We have lipids, proteins, carbohydrates. What about water? Water also is there, right? So let's write water. Water also can be found in the plasma membrane. Water molecules, which is around 20% or something. Apart from that, there is some form of complex lipid here. Can anyone tell me which is that complex lipid? Anyone? Which is that complex lipid? The complex lipid is nothing but the cholesterol, the deadly cholesterol. Cholesterol is good for your health, but sometimes cholesterol can become very deadly also. But cholesterol can also be said here. Cholesterol. Okay. Cholesterol. So all the things make up a cell membrane. Lipids, proteins, carbohydrates, water and cholesterol. Okay. What is missing? Yes, water is missing. Apart from that, cholesterol also is missing. Okay. So hopefully bad cholesterol also is away from your life. Okay. A major lipid are phospholipid, PYQ alert, PYQ. So in lipids, which is the main lipid? Phospholipid. Phospholipid. Lecithin is an example which you will study in the other chapters. Lecithin in the biomolecules you study. Lecithin, right? Phospholipid. Phospholipids. Phospholipids that are arranged in a bilayer. So how are they arranged in a bilayer? Understand. This is how they'll be arranged. How will the students? Can anyone tell me how will they be arranged? They'll be arranged in a bilayer. They'll be arranged in a bilayer like this. Hopefully you're able to see. If not, I'll move around. This is the bilayer. How they'll be present. Okay, I've moved aside. The bilayer. Okay, the bilayer. Now, lipids are arranged within the membrane and with the polar head towards the outside. Can you see? Polar head towards the outside. Okay, and the hydrophobic tail. This is the hydrophobic tail. This is the hydrophobic tail. The meaning of hydrophobic is it does not like water. It is like stay away from me. Water, please stay away from me. Okay. Now, this ensures that the non-polar tail of the saturated hydrocarbon, this non-polar tail, hydrophobic, which is, one second, let me draw the structure for you. I need separate slide here actually. I need a separate slide here. I don't know how will I get a separate slide. One second, students. Let me draw here only for you. Each one. So this was the structure, right? This is the structure of a phospholipid. This is called as the polar head. Polar head. This is the non-polar tail. Non-polar tail. Non-polar tail. Okay. This polar head is hydrophilic in nature. It means it loves water. Hydrophilic. While this non-polar tail is hydrophobic in nature. Hydrophobic in nature. Okay. So this is how they are arranged. This is how they are arranged. Okay. This is how they are arranged. Did you understand now, students? Tanuja, do you understand now? This is how they will be arranged. See, let me read again. Now you'll have a better idea about it. Now let me tell you again. See here. The line. Read the line and I'll explain to you. It's very simple. See. Major lipids are phospholipids that are arranged in a bilayer. So all the phospholipids are arranged in a bilayer like this. You see, all the lipids are arranged in a bilayer like this. All the phospholipids are arranged in a bilayer like this. Bilayer. Okay. Lipids are arranged within the membrane with a polar head towards the outside. Can you see? I told you. This is the polar head. Polar head towards the outside. See, polar head is towards the outside. This is the structure of cell membrane. Structure of cell membrane. Okay, can you see here? Can you see here? Polar head is on the outside. Look at this diagram. Polar heads. All of these are polar heads. Now you can visualize it. Can you see? All the polar head are on the outside. All the polar head on the outside. Non-polar tail on the inside. Are you able to see, students, now? The non-polar tail, this is the non-polar tail right here, is on the inside. Non-polar tail on the inside. Okay. Non-polar tail is also called as what? Non-polar tail is also called as hydrophobic tail. It doesn't like water, that's why it's on the inside. Okay, that's why it's on the inside. Now understand now. Very simple this is. See this. The hydrophobic tail towards the inner part. This ensures that the non-polar tail, the inside one, of saturated hydrocarbon, that is basically the phospholipids, right, is protected from the aqueous environment. Because it doesn't like water. This particular tail does not like water. That is why it is on the inside. It is protected from the any water. Hydrophobic tail. Okay. Now non-polar tail on the inside. Non-polar hydrophobic tail on the inside. That's all. Okay. In addition, phospholipid membrane also contains certain cholesterol. See, I told you that time only I told you. There is lipid, there is protein, there is carbohydrate, there is water, and there is cholesterol. 5 items in the cell membrane. Later, biochemical investigations clearly revealed that the cell membrane also possesses protein and carbohydrates. I told you, protein, carbohydrates, lipids, cholesterol, and water. Okay. The ratio of protein lipid varies considerably in different cell types. In human beings, the membrane of the erythrocyte, that is RBC, is approximately 52% protein, 40% lipid. 40% is the lipid. Okay. 40% is the lipid. Depending upon the ease of extraction, see, this is very important. Now, students, for example, now, imagine this is a protein. Imagine this remote right here is a protein in front of me. Okay, this right here is a protein. This one. Now I am keeping it inside my hand. I am tightly holding it. Can you remove it now? Can you remove it? If I am tightly holding it on the inside, will you be able to remove it now? No. You can't remove it at all. Very difficult to remove. Now what if I am holding in the corner and I have given you this much space to hold and remove it? Can you remove it easily now? Yes. In this case, you can remove it very easily. If I hold it on the inside completely, you cannot remove it. So based on the location and the ease of extraction, how easily you can remove the protein out. There are two types of proteins based on that. Ease of extraction. That is called as ease of extraction. Ease of extraction. So based on the ease of extraction, students, how easily you can extract, we have something called as integral proteins. We have something called as integral. Integral means what? Completely inside. Integral proteins will be completely inside. You can't remove it. The other type of proteins is called as what? Other type of protein is called as peripheral proteins, which are present on the outside. Peripheral proteins which are present on the outside. That you can remove easily. See, imagine peripheral protein is present on the outside, you can remove it easily. Integral protein which is already on the inside, you cannot remove it. Integral, you cannot remove it at all. Okay. See here. Depending on the ease of extraction, that is called as ease of extraction. It's called as ease of extraction. Membrane proteins can be classified into integral and peripheral. Peripheral proteins lie on the surface of membrane, while integral proteins are partially or totally buried in the membrane. See the diagram and understand. See here, do you see, students? We have integral protein here, which is completely on the inside. Then we have peripheral protein here, which is on the outside. This peripheral protein can be easily be removed. But this integral protein cannot be removed. Integral protein cannot be removed. That is the difference. That is the difference here. I hope everyone understands the difference between integral as well as your peripheral protein. Okay. This is the structure. Can you see? Sugar is your carbohydrates, lipids, right? Cholesterol, everything is present. Everything is present. Now, an improved model of structure of cell membrane was proposed by Singer and Nicholson in the 1972, widely accepted as fluid mosaic model. Fluid mosaic model was given by Singer and Nicholson in the 1972. According to this, the quasi-fluid nature of lipid. Quasi-fluid means what? Quasi-fluid means semi-fluid nature. Semi-fluid nature. Semi-fluid nature of lipid enables lateral movement of proteins within the overall bilayer. So students, understand there are two types of movements now. Understand? Two types of movements. One is called as flip-flop movement. Flip-flop movement. Other one is called as lateral movements. Lateral movements. Okay. This lateral movement is shown by lipids and proteins. Lipids and proteins. Then there is something called as flip-flop. Flip-flop is only showed by lipids. Only showed by lipids. Now what is the difference between flip-flop and lateral? Now understand. What happens in flip-flop first of all? Flip-flop. First flip-flop. I'll show you flip-flop first. This is the lipid bilayer, right? This is the lipid bilayer. This is the lipid bilayer. In flip-flop, this lipid will go this side and this lipid will come this side. Complete flip-flop. It is happening between the two layers. This is happening between the layers. Can you see? between the bilayer. Between the bilayer. One lipid which is on the top will come on the bottom. This lipid from the bottom will go on top. That is called as flip-flop movement. Understood? Flip-flop movement. Now what is lateral movement now? Lateral movement is very simple. This particular lipid will go this side. This will go this side. That's all. Lateral movement. Here to here. Lateral movement will happen within the membrane. It will happen within. Can you see? Only one membrane is involved. In lateral movement, only one membrane. This is called as lateral. Right? This is happening within the membrane. Within the one membrane. Monolayer. It is mono. Only one membrane is involved. Lateral movement happens. Imagine we have a protein here, students. Can you go back? See. This protein, integral protein right here. This integral protein right here can move this side, or it can move this side. Lateral movement, because it is semi-fluid in nature. Entire membrane. Understood? Now, proteins within the overall bilayer. This ability to move within the membrane, see, within, for lateral, is measured by its fluidity. So we can tell your entire plasma membrane is semi-fluid. Why? Because the structures can show movement there. Lateral movement and flip-flop movement. Okay. The fluid nature of membrane is also important from the point of view of functions like cell growth. See, this is a very important functions of plasma membrane. Cell growth, formation of intercellular junctions, secretion, endocytosis, and cell division. The functions of cell membrane. Now, why is all these functions are happening? Because the membrane is quasi-fluid in nature, or can I say it is semi-fluid in nature. If something is very semi-fluid, things can move in that. Understood, all of you? The importance of semi-fluid. Importance of semi-fluid nature. Okay? It's very easy to understand. Now, one of the most important functions of plasma membrane is the transport of molecules across it. All of you know, we call plasma membrane as selectively permeable. If I call it selectively permeable, or it should be able to transport some things. Okay. Now I'll give you a simple analogy. Okay. You're going from your house to your friend's house. Your dad or your mother is dropping you. Do you have to pay money to your dad or mom to drop to your friend's house? Tell me. Do you have to pay your dad and mom money? No. Free of cost. Aram se you can go and come. You know your parents, you know your mom, you know your dad, they will not take any money from you. Now if you go from your house to your friend's house, but you're going in a cab, you're going in an auto, taking a bus, taking an aeroplane, taking one train. Okay. You're taking aeroplane and all, do you have to pay the fees? Yes. When you have to pay the fees, it is called as active transport. When you don't have to pay the fees, it's called as passive transport. Understand again one more time now. The membrane is selectively permeable by some molecules present on the either side of it. Many molecules can move briefly across the membrane without any requirement of energy, without any money you're traveling. And this is called as passive transport. Okay, without any money. Neutral solutes may move across the membrane by process of simple diffusion. They can simply go along the concentration gradient, that is from higher concentration to the lower concentration, osmosis. Right. Water may also move from this membrane to the higher, from a higher to lower concentration. See, from higher to lower you can easily flow. Higher to low, water can flow. Okay. Doesn't require any courage. No money is required at all. Right. Now, movement of water by diffusion is called as osmosis. That is movement of water molecules from the higher concentration to the lower concentration. Doesn't require any money. Okay. Or energy. As polar molecules cannot pass through the non-polar bilayer, they cannot pass through. Polar molecules cannot pass through. Okay. They acquire a carrier protein of membrane to facilitate the transport. So what do? Some polar molecules, since polar molecules cannot go on their own, polar molecules will form a bond with certain carrier proteins. Polar molecules, right? plus carrier protein of the membrane, and then they will enter the membrane. Okay, that very important term. Okay, a few ions or molecules are transported across the membrane against their concentration gradient. Now, what is against concentration gradient? Against means lowest from the low to high. Against concentration gradient means low to high you're going now. Now, when you go to low to high, you need to spend energy, more energy. You're taking taxi, you're supposed to pay the money, right? From low to high, lower concentration to high concentration, such transport is called as an energy dependent process. ATP is used and is called as active transport. An example is sodium potassium pump. Basically the whole concept is when you have to go from high to low, free of cost. Low to high, you have to pay. Understood? Very simple concept. When coming from high to low, nothing is required. Imagine you are trying to run the downhill. When you're climbing down the stairs, you don't have to spend a lot of energy. But you have to, when you climb the stairs, you have to spend a lot of energy. Simple, same concept. Okay? Now, with that being said, our friends, can we start with the cell wall now? So, someone was asking me a question, right? Bacterial cell wall and everything. Now we will come to the cell wall. Okay. First of all, tell me, is cell wall living or dead? Is cell wall living or dead? Cell wall is living or dead? Quickly. Cell wall is living or is it a dead thing? Your cell wall, students. Cell wall is dead. Cell wall is completely dead. Okay. It is dead and it is completely rigid. It is dead and it is completely rigid in nature. It is completely dead and rigid. Tell me, is it outer to the plasma membrane or is it inner to the plasma membrane? Cell wall is always outside of the plasma membrane. Can I tell? Outer covering to the plasma membrane. Outer covering to plasma membrane. Outer covering to the plasma membrane. Okay? Plasma membrane. No. Plants have it. Right? Your some bacteria has it. Fungi also have it. Yes. Fungi also have it. Right? Algae also have it. All of them have it. The same. That is cell wall. Everyone has a cell wall. Okay, one thing, students. Everyone has the cell wall. Okay. Now, you need to understand functions. Tell me the function. What is the function of a cell wall? The first function will be obviously if it's the outer covering. Can I tell the main function of the cell wall is protection? Yes, protection. Okay. Second one, can I tell it provides shape? It provides shape and rigidity. Rigidity, sir. Okay. Rigidity. Can it form a barrier to make sure nothing unwanted enters inside, like a checkpoint? Yes. Right. It can prevent cell from bursting. Because of turgidity. Prevent cell from bursting. Cell enlarges because of turbo pressure. If there is no cell wall, it will blast off. So make sure the cell doesn't blast. It is containing the explosion. Okay. Prevent cell from bursting. And the last point is going to be cell to cell interaction. This I'll show you. Cell to cell interaction. This last fifth point is very important. I'll teach you today what is cell to cell interaction also. Okay, cell to cell interaction. Now, what is given here? Non-living rigid structure called a cell wall performs an outer covering to the plasma membrane of fungi and plants. Cell wall. Now why are they not given bacteria here? Because they are talking about eukaryotic cell. We're talking about eukaryotic cell right now here. That's why bacteria is not given here. That's why. Understand? Okay. Now, it not only gives the shape to the cell but also protects. I told you protection, shape, protection. from mechanical damage infection. It also helps in cell-to-cell interaction. I'll take you this diagram, I'll show you. And provides barrier to undesirable micromolecules. Algae also have cell wall made up of, very important, cellulose, galactans, mannans and minerals like calcium carbonate. Galactans, mannans, cellulose, minerals like calcium carbonate. Now what about in the case of plants? In the case of plants, it will obviously made up of cellulose, hemicellulose. Cellulose and hemicellulose and pectins. Okay. Now what about fungi? Can anyone tell me? Fungi. What is cell wall of fungi made up of? It is given in your biomolecules. Go check. Quickly go check. Tell me. Cell wall of fungi made up of? That is a good question. Cell wall of fungi. Tell me. While the other plants consist of cellulose, hemicellulose, pectins and proteins. Okay, pectins and proteins. Anyone fungi? Fungi is made up of chitin. In fungi, the cell wall is made up of chitin. Okay, made up of chitin. See, this is called as cell-to-cell interaction. Can you see? Cell-to-cell interaction. Now, this cell is able to interact with this cell. How? Because within the cell wall, there are small gaps. Okay. This middle part is called as middle lamellae. Middle lamella. Can you see? This is called as middle lamella. Okay, middle lamella, which is made up of calcium pectate. Called as middle lamella, made up of calcium pectate. Now, within this middle lamella, students, there is certain plasmodesmadal connections. Through this middle lamella. Now, what is happening in plasmodesmadal connections? In plasmodesmadal connection, there is connection between cytoplasm. So the two cells are connected to each other by the plasmodesmadal connections. What exactly is connecting here? The cytoplasm of the two cells are connected. The cytoplasm are connected here. Okay, cells are connected. Clear? Joined by plasmodesmata. Exactly. Okay. Now, students, apart from that, what should I teach you? There's something called as understand this one small concept is there. I'll move aside also if I can. I don't have space to write. I should have given more space to write actually. I think I'll erase this above part and I'll write here. I'll write here. Concept, you should understand the concept, right? Now, understand what is something called as primary cell wall, secondary cell wall. Ready? Okay. Plants, they have something called as primary cell wall, students. You see, they'll have one cell wall is there. This is called as primary cell wall. Primary cell wall will be present in the young stage of a plant. Okay, primary cell wall. Soon, this primary cell wall will be replaced by a secondary cell wall on the inside. On the inside, we have the secondary cell wall coming up on the inside. Clear? Secondary cell wall comes on the inside. If this is the one cell, this is cell wall, this is one more cell wall. 1 second, let me draw some gap should be there. This is one cell wall. This is one cell wall. We have one more cell wall here. Right, one more cell wall here. Between the two cell walls, there is a gap, right? You can see. This is called as, here we'll have middle lamellae. Here we'll have middle lamella in the middle. Okay? Which is made up of calcium pectate. See here. The cell wall of young plant, the primary wall is capable of growth. The primary cell wall is able to grow. It is much more permeable in nature. Young plants, right? Young plants are more permeable. But again, it diminishes and we get the secondary cell wall. See, which gradually diminishes and the cell matures and secondary cell wall is formed on the inner side of the cell. On the inner side, it is made. Inside, you have the secondary cell wall. The middle lamella is a layer mainly of calcium pectate which holds glue or different neighboring cells together. So middle lamella is the glue which is holding the two cells together. Can you see here? The two cells are connected with the help of middle lamella. Right. The cell wall and the middle lamellae may be transversed by a plasmodesmal connection connecting the cytoplasm of the neighboring cell. Here I told you, right? Plasmodesmal connections connecting the cytoplasm of the two neighboring cells. Middle lamella is the glue in the middle. Understood, all of you? Understood? It's the glue in the middle. Now, with that, next we will come to endomembrane system. Any doubt, quickly ask me. I look at the chart right now. I will look at the chart. Any doubt, quickly ask me. Now we will come to endomembrane system. Endomembrane system. Primary cell wall, functions. What is middle lamella? Right? Calcium pectate, in the middle of calcium pectate, very important point. And every single point is covered for cell wall. Any doubt, quickly ask me. I'll wait for 2 minutes to look at the chat. Any doubt, quickly ask me, students. Any doubt? Beautiful, no doubt at all. Now we'll come to something called as endomembrane system. So students, understand the meaning of system first of all. System. The meaning of system is that when multiple components are working together, when multiple components are working together for a common purpose. For example, computer system, digestive system, right? They're working together to make a food digest. So here also you have something called as endomembrane system. Here also something called as endomembrane system. While each membranous organelles is distinct in terms of structure and function, the members of endomembrane system may be different in structure and function, but their functions are coordinated. But functions of these considered together as endomembrane system because their functions are coordinated. Structurally they might be different, but their functions are coordinated. Okay, functions are coordinated. The endomembrane system includes what? This is a famous question, very simple. Every gamer loves victory. Every gamer loves victory. That is E for endoplasmic reticulum, G for Golgi complex, L for lysosomes, and V for vacuoles. All of these are belonging to the endomembrane system. Endomembrane system, all of their functions are coordinated and they are happily ever after. Okay, endomembrane system. Since the functions of mitochondria, chloroplast and peroxisomes are not coordinated, their functions are not coordinated at all. So they are not a part of endomembrane system. Chloroplast, mitochondria, peroxisomes, not a part of endomembrane system. Only these 4 organelles are part of endomembrane system. Why? Because their functions are coordinated. That's all. For example, in the class also you make some group, nice group. Why? Even though you might be coming from different locations, even though your habits might be different, but somehow will have that one connection, which will hold your team together, your group together, like that. This is the team which has different structures, right? Team with different structure, but they have a common function. They can not common function, they can functions are coordinated. Okay. Now, last topic for today, students. Last topic for today. Okay. Last topic for today we'll do today, and after this we will stop. Okay. Endoplasmic reticulum. In the part 2 we will do Golgi complex and rest of the things. Okay. Last part we'll do that. Now, let's understand endoplasmic reticulum. Very, very simple. Endoplasmic reticulum is very simple to understand, and here we go. Okay. Now, students, for example, if this is one cell, this is one cell. Okay. This is one entire cell. Let me draw rectangle. We have a cell here, friends. We have a cell here. We have a cell here. Okay. In this cell, if this is the nucleus, this is the nucleus in the middle. Students, from the nucleus is double membrane, right? Nucleus is double membrane. Yeah. Nucleus is double membrane structure. Nucleus is double membrane. Okay. Let me draw it properly again. So let's draw the nucleus first. Double membrane nucleus, and we have the cell. Cell is here. From the outer layer, from the outer wall, can you see this is the outer wall, right? From the outer wall of the, from the outer layer of the nucleus envelope, from the outer layer of nuclear envelope, we can see the development of ER. Endoplasmic reticulum will always develop from the outer layer of the nuclear envelope, like this. Okay, so now can you see, but the entire cell right now is divided into two parts. Entire cell is divided into two parts. A parts where endoplasmic reticulum is kind of absent here. Can you see the green part where endoplasmic reticulum is absent? There are some parts where endoplasmic reticulum is present. The blue color part where endoplasmic reticulum is present, right? This all is blue region that inside the ER. This blue region inside the ER, which is dividing the cell into two half, this part here is called as luminal. This part is called as luminal. This green color part is called as extraluminal. Extraluminal. This extraluminal part is basically the cytoplasm, right? The cytoplasm is the cytoplasm. What is the luminal part, students? Luminal part is basically the inside the ER. Inside the ER is the luminal. Okay, so inside the ER, whatever we have a blue color, that is luminal. Outside part is called as extraluminal. Okay, extraluminal. Apart from that, there are two types of ER. If a endoplasmic reticulum has ribosomes, we call that rough ER, RER, because it is ribosomes. If it is an ER plus no ribosome, it is called as smooth endoplasmic reticulum. This rough ER, students, RER produces proteins, synthesizes proteins. Okay, synthesizes proteins. While your rough ER, while your smooth ER is responsible for the release of the lipids and certain steroidal hormones. That's all. Let's read what is given here. Electron microscope studies of eukaryotic cell reveal the presence of network of reticulum. Can you see this is the network of reticulum of tiny tube structures scattered in the cytoplasm. Can you see tiny tube-like structures scattered in the cytoplasm? This is called as ER. The ER divides the intracellular space into two distinct compartments, luminal and extraluminal. Luminal is inside ER, extraluminal is the entire cytoplasm part. The ER often shows ribosomes attached to the outer surface, and the endoplasmic reticulum bearing ribosomes is called as RER. Without is called as smooth ER. Rough and smooth. Rough ER is involved in the protein synthesis, and they are extensive and continuous with the outer membrane of the nucleus. Can you see? Outer membrane of nucleus. Continuous. The smooth endoplasmic reticulum is the major site for synthesis of lipids. In animal cell, lipid-like steroidal hormones are also synthesized by SER. The steroidal hormones I am talking about is nothing but from the testosterone and ovaries. Testes and ovaries, whatever the hormones they release, testosterone and estrogen, responsible for all those hormones. Okay, so remember the function, what is luminal, extraluminal, what is smooth and what is rough. So with that, today's class is over. Okay, next class, students, can you see here? This is your rough ER, continuous from the outer layer. Then we have smooth ER on the separate. See, continuous it is. This is the nuclear membrane, nucleus, nuclear pore. This is nuclear outer layer. From outer layer is continuous from the outer layer. Next class we'll do Golgi and lysosomes, vacuoles, mitochondria, plastids, right? All of that we'll do in the next class. Lysosomes, structure of your centrioles, structure of flagella, all that we'll do in the next class. Okay. Next class probably we'll do it on Saturday. Let's do it on Saturday. Okay. So with that being said, students, thank you so much, students. Let me know in the comment section how was the session. I know it was a little dull, but we are covering every line of NCERT. We are doing justice to the class. And if you're attending the class, go try solving some questions now. After this class right now, go try solving questions on this chapter. Whatever I've taught till now, you will be able to solve every single question from whatever we have learned today. Every single question, you'll be able to solve like this. So thank you so much, students, and that's it for today. I'll see you in the next class. This is Baswaraj sir.